Even if I gave you a cell, I gave you a cell that just died, because presumably a resurrection is easier than a de novo synthesis. Even if I gave you a cell that just died, could you bring it back to life?
Well, one of the biggest areas of controversy is with, say, biological evolution. And that's been one of the centers of the culture wars between science and scripture. But distinguishing the question of biological evolution, which is downstream from you're a synthetic chemist, right? So you have something upstream before the biologist is even born, so to speak. And this is maybe we call it the question of chemical evolution or whatever. It concerns the origin of life itself, a biogenesis, or the problem of abiogenesis. You're the person who knows how this is supposed to work. So how did life begin?
Okay. As a scientist, we have absolutely no idea. Nobody has any idea. Even origin of life people who say that they have an idea, as soon as I poke them on it, they have no idea. So tell me about it. Go ahead, tell me. You like your primordial soup model where there were some molecules in a pond, flashes of lightning, these molecules come together and form a cell. Okay, that's a big step. You got to somehow take very small, simple molecules like formaldehyde and CO2, and somehow you've got to make the 4 classes of chemicals. You've got to make carbohydrates. You have to make proteins, first the amino acids, and then the proteins from those. You got to make the lipids, which people think are easy. They're not easy at all. And you have to make the nucleic acids, which is the DNA and RNA. We don't even know in any prebiotic type chemistry experiment how to make any of those classes of chemicals in a homochiral form. Homochiral meaning you get one mirror image and not the other. We know that chirality could not have come later on during an evolutionary process. You had to have near perfect chirality. And we know that just for the things to fit together, but we know that from something new in the last 20 years, chiral-induced spin selectivity. A cell would burn up if you don't have the right chirality, because you have to have chirality matching to what's called the spin of the electron. Each chiral element is a spin valve. And that's why we don't burn up. That's why we do all these computations in our head, and we're only burning 10 watts, whereas my computer is burning gazillions of watts, lots of watts. I mean, it's just, and so you see this difference. Even if you were given the homochiral compounds, and with sugars, it's extremely hard. Carbohydrates, how to do this. But even if you were given those, how do you polymerize them now to make the polysaccharides that you're going to need? Nobody knows how to do that. Everybody's clueless on that. How do you polymerize the amino acids? People, you'll say, well, wet-dry cycles. Well, wet-dry cycles don't do it for you, because you have half of the amino acids have active side chains that participate in the coupling. So you don't have the peptide bond anymore. And so you can't get those even by wet-dry cycles. And by wet-dry cycles, people have made, I don't know, hexamers maybe, tiny little things, and they're all mixed up. And so what they'll do is they'll use glycine and alanine that don't have the active side chains. Well, you load up the system, but that's not real. You want to take the 20 amino acids, it doesn't work because half of them have active side chains. So we don't know how to polymerize those. We don't know how to polymerize the nucleotides. People say, oh, it was just recently this work by Benner and Biondi from Florida, where they took pure nucleotide triphosphate that they bought from a manufacturer, pure, and they put it on these rocks, and they got some oligomerization to what they claim as high as 200 mers or 300 mers. But then when you start looking at how it connected, you have the 2' 5' linkage, not just the 3' 5' linkages, and you have branching therefore. And we know the 2' 5' linkage can't transcribe to a protein structure. And they'll say, well, a good percentage of it is the 3' 5'. Well, what percentage? Anything that's been done on a surface has anywhere from 30% to 80% of the 2' 5'. So if you have even just 30%, we'll go with the 30% number. That means 1 out of every 3 of your linkages doesn't work. Well, you need 3 linkages. You need 3 letters to define 1 amino acid in a protein. So that means you still don't have any words. You don't have any words, even with all these grand experiments. And you poke them on it, and they have no answer to this. And I poked Benner on this just recently without any answer. These things just fall apart. The problem is that people who don't know much will hear this quote from this person and think it's there. No, we don't know how to polymerize these. But even if I gave you all the polymers, I'll give you all the polymers, and I'll even give you the informational code, which we have no idea where it came from, because you need the code to prescribe the enzymes that are going to build a biological system. And I'll give you all the peptides, all the carbohydrates hooked together in any order that you want, which is an amazing test to try to figure out how that could have ever happened. But I'll give it to you. All the components of a cell. Could you put a cell together in your modern lab, let alone under a rock or in some pool someplace? Even if I gave you all the components, no, I can't put a cell together. And you say, well, haven't people made synthetic cells? No, they're not. They call it synthetic cells, but they took an existing cell and they knocked out some of the genes and they said, here's a synthetic cell. Or they took some other genome and they put it into an existing cell. Even if I gave you, and I even put this before Benner, just in last month, I said, even if I gave you a cell, I gave you a cell that just died, because presumably a resurrection is easier than a de novo synthesis. Even if I gave you a cell that just died, could you bring it back to life? I don't even know how to define what it is that I just lost. I don't even know how to define the life there. That's how far we are from this thing. And you say, well, we're right on the edge. We're not right on the edge. Jack Sostek in 2014 said in 3 to 5 years, we'll have life in the lab. Well, that was in 2014. How you doing, Jack? Now he's just trying to get the nucleotides put together. It's just not there, because what happens is we're getting further away from this. The target is moving further away from us than we are approaching it. Because what happens is we learn more about the complexity of a cell. We learn about chiral-induced spin selectivity in the last 20 years. That I got to have all the chirality in place. I don't even know how to do this. Even if I had it, how do I get the spin of the electron to match with this thing? We don't know how to do this. That's called and then there's the interactome. The non-covalent interactions are very important in a cell. The non-covalent, just this is how molecules line up with each other. Just in a single yeast cell, the non-covalent interactions of just protein are 10 to the 79 billion power combinations. The number of elemental particles in the universe is 10 to the 90. This is 10 to the 79 billion. This is crazy big number. How do you get these to line up? And you got to have the right arrangement in order for information to transfer. That's just protein in a simple yeast cell, let alone a biological cell like a human cell. So all of these, that's called the Leventhal 2.0 paradox. All of these are mysteries that we didn't even know of a couple of years ago, or a couple of decades ago. So the target gets further away from us. And so we are getting further from solving this than we are from solving it. So that's the origin of life. Without the origin of life, you can't have evolution, because evolution is from that first cell to where we are today. I can deal with that issue as well, but there's the origin of life.